Welcome to Bright Scientists. We are hoping to inspire, motivate, and educate you young scientists and help you to become even brighter scientists. I hope you enjoyed my last video on cells and organisation. If you haven't had a chance to see it yet, I will post a link in the description below. Without you guys, we wouldn't be able to grow this page, so continue to like, comment and subscribe and share the word of Bright Scientists. Today's lesson we'll be looking at microscopes and how they are so useful to look at microorganisms. According to the national curriculum, you need to be able to understand what the different parts of the microscope are and how to calculate magnification. I don't know whether you know, but the earliest microscopes were often known as flea glasses because they were used to study small insects as they used to magnify them and make them look bigger. Why not check out this news article on microscopes and how they won the Nobel Prize in Chemistry? So moving on, as you can see, there is a notepad, so how about you jot these ideas down? Microscopes are used to look at objects too small for the naked eye. Microscopes are made up of magnifying lenses which make the specimen look bigger. Over here you can see that there are images of different organisms under a microscope. We have the hydrothermal worms, the mealworms, which are used to magnify their faces and reveal its eyes and mouth parts really clearly. They are the larval form of the mealworm beetle and are commonly used as high-protein feed for the animals. The next image is the breeding Daphnia, often used at A-level biology to look at their heart rate, as you can see the structures clearly under a microscope, and also amoeba. In your exam, you will need to know the definition of magnification, which means enlargement, and resolution, which refers to the image clarity. There are two types of microscopes that you need to know about, which is the light microscope and also the electron microscope. Most student microscopes are classified as light microscopes. These are the ones that you see in school. This is because in a light microscope, visible light passes through the specimen, the biological sample you are looking at, and is bent through the lens system, allowing the user to see a magnified image. However, if you want a higher resolution microscope, then electron microscopes are better and are often used in the medical field. Electron microscopes differ from light microscopes in that they produce an image of a specimen by using a beam of electrons rather than a beam of light. Electrons have a much shorter wavelength than visible light, and this allows electron microscopes to produce higher resolution images than standard light microscopes. Electron microscopes can be used to examine not just whole cells, but also the subcellular structures and compartments within them. So, let's now take a look at the parts of a microscope. We have all these different parts of the microscope that we are going to explore further in the next slide. So make sure you pause the video now to try and research why we have two focusing knobs. You're correct. The answer is because we need two functions, one to bring the specimen nearer and the other one is to focus the image. So let's look at the parts of the microscope. The eyepiece, also known as the ocular. This is the part used to look through the microscope and is found at the top of the microscope. We have the handle, in which you can hold the microscope, and we also have two focusing knobs. One, which is the coarse knob, which is used to bring the specimen into approximate or near focus, and we also have the fine focus knob, which is to sharpen the focus quality of the image after it has been brought into focus with the coarse focus knob. We have a microscopic illuminator, which is the light source located at the base of the microscope. It is used instead of a mirror. It helps to capture light from an external source or a low voltage of around 100 volts. We also have a stage in which the specimen is often placed. Usually we have the stage clips which hold the actual specimen in place. And we have the objective lenses, which is used for the specimen visualization, and it's used to help magnify at around 40 to 100 times. Pause the video now and try to research more about the rough and fine focusing knobs. We now need to think about how do we actually use a microscope. So, first of all, you need to carry your microscope by the handle and place it on a flat surface. You then need to make sure that your microscope slide should be prepared and also have a cover slip placed over the specimen. You need to make sure you place the slide onto the stage and fasten with stage clips. And turn the light microscope on and adjust so light shines through. You then need to select the lowest objective lens on the microscope, so it can usually be 40 times. You turn the rough focus knob to move the object lens down to just above the slide. And next you look through the eyepiece and the focus using the fine focus knob. Keep adjusting till you see a clearer image. We now need to know how to calculate total magnification. So, I know there are a load of you mathematicians out there. So, in order to calculate total magnification, we need to have an equation. We need to do the eyepiece lens times by the objective lens in order to calculate total magnification. So, in this example, we have an eyepiece lens of 15 times, and we have an objective lens of 10 times. So what would the magnification be? Pause the video now and have a go. For the challenge question, we also have a different type of question. We have been given the total magnification of 260 times, and we have an eyepiece lens of 15 times. So now calculate what is the objective lens. For this one, you would have to do division. Get a green pen out now, as we are going to mark the answers. You should have got 150 times and also 17.3. You get 17.3 as you do 260 divided by 15, which gives you 17.3 times as the magnification. So always, the key message is to make sure that you remember your equation. Right. You now need to pause the video and think about comparisons between a light and electron microscope. This is always an exciting exam question that always comes up, so make sure you revise this. So, common things that we need to look out for. Light microscopes only use light. They have lower magnification, lower resolution. The image that is often produced is a 2D image. It's also often cheaper and can be found in school settings, and it's also portable. However, in an electron microscope, it uses electrons, it has a higher magnification, a higher resolution. That means it also produces a 3D image. And it's often expensive as it's used in laboratories. And as you can see, it's a really big instrument, so it's hard to move. One thing that is important for both of them is that they both are microscopes, which are used to look at specimens, and they both contain lenses. So this is the key information that you need to take from this slide. So let's try an exam question. This question has 7 marks. So I'd like you to pause the video for 7 minutes now and try this exam question. So we're now going to mark with green pen. The first question asked, which part of the microscope can be adjusted to allow light in? That would be the mirror. The next question said, how would you make the image of an onion cell appear bigger? So what you would do is you would switch the objective lens to one which is higher powered. So as you saw, we went from 40, then there was one to 100. You would then refocus the microscope to look at the image clearly. It then said, what's the difference between light and an electron microscope? So light microscopes use light, whereas electron microscopes use electrons. You can also say that light microscopes have a lower magnification and lower resolution compared to an electron microscope. Light microscopes give a 2D image, whereas electron microscopes can give 3D images. You can also talk about how light microscopes are often used in schools and so are therefore cheaper than electron microscopes which are used in hospitals, for example. I'd like to say now, thank you for watching this YouTube video on microscopes. If you enjoyed this video, please like, comment, share and subscribe to this channel. I can't wait to continue creating content for you guys. Remember, make the leap to become brighter scientists.